You're sitting in a waiting room somewhere. Fluorescent lights, a magazine from 8 months ago, the faint sound of a TV in the corner playing something nobody asked for. The person next to you is scrolling their phone. The person across from you is doing the same. You glance down and realize you're doing it too. 3 humans in a room together, in complete silence, each staring at a glowing rectangle held at exactly the angle that strains the neck just enough to not quite hurt yet. And somewhere in the back of your mind, unprompted, a thought surfaces. Is this doing something to us? Not in the vague parental concern way, in the actual biological, encoded into the genome way. Are we, right now, in real time as a species, changing? Not culturally, not technologically, physically, genetically. Are we becoming something our great-great-grandparents wouldn't quite recognize as the same animal? The answer is yes. Unambiguously, measurably, sometimes absolutely bafflingly, yes. And the ways it's happening are strange enough that if you wrote them into a science fiction novel, an editor would probably ask you to tone it down. First, we need to demolish a myth that has a surprisingly tenacious grip on popular understanding. The idea that humans have stopped evolving. This idea comes from a reasonable sounding place. Evolution, as most people understand it, requires selective pressure. Environmental challenges that favor certain traits, killing off individuals who lack them before they reproduce, thereby changing the frequency of those traits across generations. The reasoning goes, modern medicine, agriculture, and technology have removed those pressures. Traits that would have been lethal in the wild are now survivable. Nobody is dying from nearsightedness anymore. Nobody is being outcompeted reproductively because they have flat feet or a weak immune response. Therefore, the story goes, selection has relaxed and evolution has stalled. This is wrong in almost every interesting way a thing can be wrong. Natural selection is not the only mechanism of evolution. Genetic drift, the random fluctuation of gene frequencies across generations, independent of survival advantage, operates continuously regardless of medical technology. Mutation rates have not changed. Gene flow between populations, historically constrained by geography, has accelerated dramatically as human migration increased. Sexual selection, who chooses to reproduce with whom and why, is as active as it has ever been and operates under completely novel cultural pressures. And natural selection itself hasn't disappeared. It has changed shape. Modern environments create new selective pressures, some subtle, some surprisingly strong, and the human genome is responding. We know this not philosophically but empirically. Large-scale genomic studies comparing populations across recent centuries have detected signatures of selection, allele frequencies shifting in ways that cannot be explained by chance alone, across dozens of traits. Some of these are expected. Some of them are lie bizarre. Start with something almost funny in how mundane it is. The wisdom tooth. Wisdom teeth, third molars, are vestigial. They were useful when ancestral human diets required serious grinding power, and when early tooth loss from wear and decay meant the back of the mouth had space to fill. Modern diets don't require that grinding capacity. Dental hygiene has improved tooth retention, and jaw sizes have trended smaller over time as dietary texture softened. The result, wisdom teeth now arrive into a jaw that frequently doesn't have room for them, causing impaction, pain, infection, and a thriving market for oral surgeons. But here's where evolution starts catching up in real time. A measurable and increasing percentage of the human population is now being born without wisdom teeth at all. The gene variants associated with wisdom toothogenesis, developmental absence, have been increasing in frequency. In some populations, particularly in parts of Asia, rates of complete third molar absence now exceed 30 to 40%. This is not random variation. The pattern of increase, the population distribution, and the association with another trait, an extra artery in the forearm called the median artery, all suggest active selection or developmental linkage. Speaking of which, the median artery is one of the more remarkable recent discoveries in human evolutionary biology, and it deserves more attention than it typically gets. During fetal development, all humans initially form a median artery in the forearm, a vessel that supplies blood to the hand while the limb develops. In most people, this artery regresses as development proceeds and is replaced by the radial and ulnar arteries, which take over as the permanent supply. By adulthood, in most individuals, the median artery is gone, except it increasingly isn't. A 2020 study examining cadavers across several decades found that the proportion of adults retaining a persistent median artery has increased significantly over the past century and a half. In cadavers from the late 1800s, retention rates hovered around 10%. In cadavers from people born in the mid-20th century, that figure had climbed to around 30%. The researchers projected that if the trend continues, the persistent median artery may become the norm rather than the exception within a few centuries. Why? The honest answer is that nobody is certain. One possibility is that the genetic variants responsible for median artery persistence are being positively selected, perhaps because an extra blood vessel in the forearm provides some marginal circulatory advantage, or perhaps because those variants are genetically linked to something else that is being selected. Another possibility is that developmental processes are shifting in response to environmental factors, producing retention as a byproduct. But regardless of mechanism, the trajectory is clear. Humans are, right now, in the process of acquiring a new persistent artery. An anatomical structure is being added to the standard human blueprint. This is evolution happening in a time frame we can actually observe. It is extraordinary, and it happened in a body part you use every day to hold your phone in that waiting room. Now let's go somewhere that feels more socially uncomfortable, because evolution doesn't have a sensitivity filter and doesn't care about our discomfort. Lactase persistence, the ability to digest lactose, the sugar in milk, past infancy, is one of the most studied and most dramatic examples of recent human evolution. Most mammals, including most ancestral humans, produce lactase only during infancy. After weaning, the gene responsible for lactase production switches off, and the ability to digest milk fades. Consuming significant quantities of fresh milk as an adult would then result in the bloating, cramping, and digestive misery that lactose-intolerant people experience today. Then, roughly 10,000 years ago, in multiple independent populations that had begun keeping cattle, something changed. Genetic variants that kept the lactase gene switched on into adulthood began to spread. In populations with long histories of pastoral dairying, Northern Europeans, certain East African groups, some Middle Eastern populations, these variants spread so rapidly and so completely that they now approach near fixation. In Northern European populations, lactase persistence rates exceed 80%. This is one of the fastest documented instances of natural selection in human evolutionary history. The selective advantage was enormous. In environments where fresh milk was a reliable caloric resource, individuals who could exploit it had better nutrition, better survival odds, better reproductive success. The genes spread like wildfire through those populations. Meanwhile, in populations without that pastoral history, much of East Asia, most indigenous populations of the Americas, many sub-Saharan African groups outside cattle herding traditions, lactase persistence rates remain low. These populations didn't experience the same selective pressure, so the variants didn't spread. This is not a medical condition in any meaningful sense. It is two different evolutionary trajectories, running in parallel within the same species, responding to divergent cultural practices. Humans domesticated cattle, and cattle domesticated our genomes right back. Here is one that hits harder than most people expect. Humans are currently evolving in response to modern medicine and healthcare. The argument that medicine removes selective pressure is, on close examination, precisely backwards in some domains. Medicine doesn't just allow previously lethal conditions to survive, it changes the reproductive landscape in ways that can accelerate genetic change in particular directions. Consider the evolution happening around the BRCA gene variants associated with elevated breast and ovarian cancer risk. These variants have likely always been present in human populations at low frequencies. Historically, their effect on reproductive success was limited by the fact that the cancers they predisposed toward typically manifest after reproductive age, meaning selection couldn't act on them efficiently. Modern medicine, by enabling diagnosis and preventive intervention, has changed this calculus. Simultaneously, the psychological and social responses to known BRCA status, including decisions about reproduction, family size, and timing, represent a novel selective environment that has no analog in human evolutionary history. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. More broadly, traits associated with surviving or thriving in healthcare-dependent environments are now under selection in ways they weren't previously. The genetics of immune function are shifting in populations with high antibiotic exposure. Variants affecting drug metabolism, how quickly the body processes specific pharmaceuticals, show patterns of regional differentiation consistent with selective pressure in populations with different medication histories. We are evolving in response to our own medical interventions. The species is co-evolving with the civilization it built. The genetics of reproduction itself are changing in measurable ways, and this is where things get philosophically interesting alongside the scientifically fascinating. Age at first reproduction is a trait with a heritable component, meaning genes influence in part when individuals tend to start having children. In modern Western populations, strong cultural pressures push reproduction later. Education, career development, economic factors, social norms. These factors delay reproduction for many individuals, but the heritable component means that individuals whose genetic predispositions lead them toward earlier reproduction, independent of cultural pressure, will, on average, have more children than those whose predispositions push later. Over enough generations, this creates directional selection toward earlier reproductive tendency, even within populations where the cultural norm is late reproduction. The people bucking the cultural trend most successfully in reproductive terms are disproportionately passing on their alleles. A large study using data from the Framingham Heart Study cohort tracked this directly in a real population over several generations and found exactly this signal. Women with genetic predispositions towards slightly earlier first birth and slightly more children were, in fact, incrementally increasing in frequency in the population. The effect size per generation was small, but it was there. It was consistent, and it was in the direction selection theory predicted. This is evolution operating in a modern Western democratic society with full access to contraception and reproductive choice. It doesn't require anyone to be dying. It requires only that reproductive success, the actual currency of natural selection, differs between individuals for heritable reasons. Here's a direction of human evolution that tends to produce a specific flavor of existential unease. We may be evolving in response to psychological and social environments in ways that are altering cognition and personality at a population level. Schizophrenia, bipolar disorder, and certain other psychiatric conditions have significant heritable components and present an evolutionary puzzle. If these conditions reduce reproductive fitness, why haven't the associated variants been purged from the population over millennia? One compelling answer is that the very same genetic variants associated with elevated risk for psychiatric conditions are also associated with traits that may carry advantages: enhanced creativity, heightened pattern recognition, greater capacity for abstract thinking, extreme sensitivity to social nuance. The hypothesis is that these variants persist because in moderate expression or in particular environmental contexts, they confer benefits that offset the costs. The same alleles that, in one configuration or developmental context, contribute to psychosis may, in another, contribute to the kind of divergent thinking that produces art, religious experience, innovative problem-solving, and charismatic leadership. If true, this means that these cognitive traits we associate with human cultural achievement, creativity, spirituality, abstract reasoning, social complexity, may be partially anchored in genetic architecture that carries psychiatric risk as a shadow cost. We evolved our inner lives and our inner chaos from the same source. And the environment we've built, urban, cognitively demanding, socially complex, stimulation-saturated, may be applying novel selective pressure on this architecture right now in ways we don't yet have the longitudinal data to fully characterize. Body size and skeletal structure are changing too, in ways that are partly evolutionary and partly developmental, and separating the two is tricky. Average human height has increased substantially over the past 2 centuries in most developed nations, primarily through improved nutrition and reduced childhood disease burden rather than genetic change. But within that environmental signal, genetic selection on height-associated variants is detectable. Different populations show different trajectories, and the interaction between genetic predisposition and developmental environment means that the phenotype, the actual body, is being sculpted by both forces simultaneously. Skull shape has been shifting. Studies of European skulls across several centuries show measurable changes in cranial form. Broader, more rounded brain cases, slightly flatter faces. These changes partly reflect dietary shifts reducing the mechanical load on the jaw and facial skeleton during development, and partly reflect shifts in the underlying genetic architecture of craniofacial development. The face your ancestors wore was shaped differently, and not just because of whatever happened at their last haircut. Bone density patterns have shifted too, in directions discussed in other contexts, but worth noting here. The skeletal robustness of pre-agricultural humans is no longer the norm, and while lifestyle accounts for much of this, the underlying genetic predispositions for bone development are also under selection in populations with dramatically reduced physical loading environments. There are stranger corners of human evolution happening right now that don't get enough attention. Take epigenetics. Epigenetics refers to changes in gene expression, which genes are turned on or off, and to what degree, without changes to the underlying DNA sequence itself. These changes are influenced by environment, diet, stress, chemical exposure, behavioral patterns. And crucially, some epigenetic modifications appear to be heritable, passed from parent to offspring, and sometimes persisting for multiple generations. This is Lamarckian inheritance, basically. The idea that acquired characteristics can be passed down, which was supposedly debunked in the 19th century and replaced by Mendelian genetics. It turns out the story is more complicated. Certain environmentally induced epigenetic marks survive the usual erasure process that occurs during reproduction and appear in offspring who never experienced the original environmental trigger. Studies in rodents have shown that stress responses, dietary patterns, and even specific learned fears can be transmitted epigenetically to offspring. The evidence in humans is accumulating. Children and grandchildren of populations that experienced famine show metabolic profiles influenced by their ancestors' starvation, even when they themselves have had adequate nutrition their entire lives. Children of trauma survivors show altered stress hormone regulation consistent with epigenetic inheritance of trauma-induced changes. If this mechanism is operating at scale in human populations, and the evidence increasingly suggests it is, at least for certain traits, then the environmental experiences of living humans are being encoded not just in culture and behavior, but in the biological inheritance we pass to our children. The modern world, with its particular stresses, its nutritional environment, its chemical landscape, its psychological pressures, may be shaping human biology across generations in ways that bypass traditional genetic change entirely. We are writing something into our descendants' biology in real time. We just can't read it yet. Perhaps the most jaw-dropping frontier in current human evolutionary biology is what's happening with our relationship to microbiomes. The human body hosts somewhere in the range of 38 trillion microbial cells, bacteria, fungi, archaea, viruses, most of them in the gut. This microbial community is not a passive passenger. It influences immune development, metabolic function, neurological signaling, mood regulation, and susceptibility to a range of conditions. The relationship between host genome and microbiome is so intricate and so consequential that some researchers argue the human organism should properly be understood as a holobiont, a composite of human cells and microbial cells that evolved together and function as a unit. Human genetic variants influence which microbial species colonize the gut successfully and persist. Different individuals with different genotypes host meaningfully different microbial communities, and those microbial communities have their own evolutionary dynamics, selection, competition, adaptation, occurring across timescales far faster than human generational time. This means human evolution and microbiome evolution are coupled. Changes in human lifestyle, diet, antibiotic use, sanitation, birth practices, alter the microbiome, which alters the selective environment for human genetic variants, which alters gene frequencies, which alters the microbial niche in an ongoing feedback loop. The microbiome of a 21st century urbanite is measurably different from that of a hunter-gatherer in ways that appear to be driving health divergences, and those differences are, in part, products of evolved differences in host genetics. We are co-evolving with 1 trillion organisms living inside us, and those organisms are adapting to the changes we make to our environment faster than we can track. Sexual selection in humans has always been complex, culturally mediated, and therefore evolutionarily unusual. But the modern environment has introduced selection pressures that have no evolutionary precedent whatsoever. Dating apps, social media, and the algorithmic mediation of mate choice mean that physical and personality traits are being filtered through technological systems at population scale in ways that never existed before. Whether this is producing detectable genetic change is too early to say with certainty. But the principle is solid. If technology-mediated mate selection consistently favors particular heritable trait profiles, and there is evidence that it does, at least at a phenotypic level, then those traits will increase in frequency over time. More immediately measurable, sperm quality metrics have declined significantly in Western male populations over the past several decades. Sperm count, motility, and morphology have all trended downward in ways that correlate with environmental chemical exposure. Endocrine-disrupting compounds found in plastics, pesticides, and industrial chemicals. These aren't genetic changes yet, but they represent a chemical environment actively interfering with reproductive biology in ways that, over long timescales, could create selection for resistance variants, or could create population-level fertility constraints that reshape who reproduces successfully. The chemistry of modernity is not neutral with respect to reproduction, and reproduction is the only thing natural selection actually cares about. Here's a synthesis point worth dwelling on. Human evolution has not slowed. The claim that we've escaped evolution is a story we tell ourselves that flatters our sense of control over nature without being true. What has changed is the landscape of selection. The pressures are different. They operate on different timescales, through different mechanisms, in different domains. Some ancient pressures have been reduced. The pathogens that once drove enormous selective sweeps are largely controlled. The nutritional deficits that stunted ancestral populations are less common. The physical attrition of subsistence life that shaped bone and muscle genetics is mostly gone from industrialized societies. But new pressures have arrived in their place. The chemistry of industrial civilization. The dietary environment of processed food. The reproductive landscape mediated by technology. The psychological environment of information overload and chronic stress, the microbial disruption of antibiotics and sanitation, the developmental environment of children who grow up indoors. Each of these is a pressure, each operates on heritable variation, each changes over generations, which variants thrive and which ones don't. The genome is listening, always, to the environment. Not with intention, not with direction, but with the relentless arithmetic of differential reproductive success that has been running uninterrupted since the first cell divided in a primordial ocean. There's something worth sitting with here, beyond the science. When you look at the evidence, the median artery spreading through the population, wisdom teeth vanishing from genomes, lactase persistence running its course in pastoral populations, epigenetic marks from famines echoing through grandchildren, microbiomes co-evolving with host genetics in real time. You're looking at something ancient and completely indifferent to human plans. Evolution does not care about civilization. It doesn't care about medicine or technology or the very human desire to believe we've transcended biology. It cares about one thing: which variants end up in the next generation and in what proportions. Everything else is downstream of that arithmetic, and the arithmetic is running right now. In you and everyone around you, and the children being born today who will carry allele frequencies we haven't measured yet into a future we can't predict. The person next to you in that waiting room, scrolling their phone with their neck at that particular angle, they are a temporary expression of a genome that is participating, without their knowledge or consent, in one of the longest-running experiments in the history of the planet. So are you. The question posed at the start was, are humans still evolving in bizarre ways? And the honest answer, padded out across everything we've covered, is yes, constantly, in ways that range from the quietly predictable to the lie unexpected to the philosophically destabilizing. An extra artery, disappearing teeth, dairy tolerance as cultural coevolution, epigenetic inheritance carrying the chemical stress of one generation into the biology of the next. Microbiomes and host genomes in an evolutionary conversation we're only beginning to translate. Reproductive selection operating through algorithms, the genome responding to the particular, unprecedented, chaotic pressures of 21st century life as faithfully as it ever responded to glaciers, predators, and famines. None of this is happening at a pace you'd notice in a lifetime. Evolution laughs at human timescales, but the fossil record and the genomic record both confirm that small, consistent, directional changes compound into profound transformation across centuries and millennia. The differences between you and a human 10,000 years ago are not trivial. Not in skull shape, not in metabolic function, not in disease susceptibility, not in the persistence of developmental structures. And the humans 10,000 years from now will look back at our genomes the way we look at the bones of our ancestors, with curiosity, with recognition, and with the quiet understanding that something was always in the process of becoming something else. We are not a finished product. We are a draft with an extremely long revision cycle. The waiting room fluorescent light is probably flickering somewhere. Your neck still hurts from that angle. The magazine is still 8 months out of date, and the body holding the phone, scrolling without thinking, is doing what bodies have always done. Participating in a process vastly older and more patient than any idea it has ever had about itself. Evolving, quietly, without asking permission, right now.